How's it going? Today we're going to talk about the processes of transcription and translation. Overall, these are the processes where DNA makes proteins, ultimately. Overall, there's two different steps for this protein making process to happen. The first one is called transcription, and the second is translation. So we're going to start by talking about transcription, and that's the process where we make this thing called mRNA from DNA. The second step, translation, is when we make proteins from the mRNA made in the previous step. Alright, so let's start with transcription. In the nucleus, this is our nucleus right here, we have DNA. And if we remember before from our DNA presentation, A's in DNA will always match up with T's on the other strand of DNA. So everywhere here on my strand of DNA that I see an A on one side, I know the other side of that DNA is going to have a T. Here's an A, so here's a T. Everywhere that there's a T on one strand, I know that's ultimately going to match up with A. So now everywhere I see an A here, a T, I'm going to put an A, excuse me. G's always match up with C's. And C's always match up with G's. So we have our DNA strands. A and T always match together, and G and C always match together. Now, this DNA is stored in the nucleus, but we can't make proteins in the nucleus. And so what has to happen is we have to have this code that the DNA makes with these bases get to this guy over here, called the ribosome. And the ribosome, if you remember from way back when we were talking about the parts of the cell, the ribosome is the cell part that actually makes the proteins. So we have a code here, and we need to get it over here. But there's a problem. One of the bases, thymine, T, right here, can't get out of the nucleus. It's stuck, and it has to do with the chemistry of thymine and the chemistry of the nucleus. You don't have to pay attention to that. All you need to know is that thymine can't get out of the nucleus. But we need this code to get over here. So the way we solve that problem is we convert this code into something else called mRNA. mRNA stands for messenger RNA, and it's exactly what it sounds like. It's a messenger. It's a messenger that brings this code in DNA all the way over to our ribosome so we can make proteins. Now, can't be easy peasy like it was with strands of DNA. We got to change it up a little bit because science is like that, right? And so what happens with mRNA is that going from this code to this code in mRNA, T's will still match up with A's. But when we match A's into mRNA, get a little weird. Instead of having T's, we have something called uracil, which is the letter U. C's still match up with G's, and C's still match up with G's down here. So the only real difference is that uracil, is different. Uracil can leave the nucleus. So that's good. Now we can have our messenger RNA message over to the ribosome. Let's practice this. Up here in my strand of DNA, I have T thymine. I know that T matches up with A. So my mRNA strand is going to have an A here. My A will match up with U. So next here will be U. My C matches up with G. My G will match up with C. T's match up with A. A's match up with U's. C's match up with G's. And G's match up with C's. Good. So now I have my strand of mRNA that can leave the nucleus. That is transcription. In transcription, we transcribe a message in DNA into a message in mRNA. Now we can start talking about this process called translation. In translation, our ribosome is going to read this strand of mRNA that came from the nucleus, and it's going to read it in groups of 3 bases. Those groups of 3 bases are called codons. So a codon is a group of 3 bases. Now what happens is, in the ribosome, these 3 bases are matched up with one of these guys right here, called tRNA. And these tRNA are floating all over the cell, just like our ribosomes. Now the ribosome here looks really, really big, right? Because we want to see what's happening in it. But in actuality, if our nucleus was this big, our ribosome would be like this teeny tiny little speck right here, right? So here's our big, giant, blown up ribosome, and here's these big, giant, blown up tRNAs floating around our ribosome. These tRNAs have 2 parts on them. This part right here is called an amino acid. An amino acid is a building block of protein. So these amino acids combine together to make proteins. The other thing in a tRNA is the anticodon. So the tRNA is transferring 2 things. It's transferring, which stands for tRNA, an anticodon and an amino acid. This anticodon is a really important thing because this anticodon is what is going to match up with this mRNA codon. So I can see that this tRNA has a C, an A, and a U, which will match up with my mRNA strand. Because my tRNA anticodon has to have the letters that will match up just like we've been doing all along, but it's going to be out of RNA. So here's my anticodon, and it's going to be attached to my tRNA. And at the top of my tRNA, I have an amino acid. And it's not just going to be any amino acid, it's going to be a very specific amino acid. And the way you figure out which amino acid it is by looking at a chart kind of like this one. So this is an amino acid wheel that tells you which amino acid is going to be going with which mRNA strand. So this is how you read it. In our codon, we have 3 letters. Here's the first one, the second one, and the third one. On our wheel, there's 4 big letters in the center. The first letter is going to correspond with those center letters. So the first letter, G, I'll find in the middle of our codon is right here. Now when I go up a row or go out a row to look at the second letter, I'm not going to look at the second letters in any part after these inner letters. So I'm just going to be looking at, not here, just these 4 letters after the G for the second letter. Our second letter is U, so I'm going to start with G was the first one, and then I'm going to go out to U. Now that I went to out to U in the second row, I can't look at any of these letters in the third row that aren't matching up with the U. My last letter is an A, so from U I'm going to move out to A. And if we look at G, U, and A, we'll see that that corresponds with the amino acid valine. Now, let's do this again. The tRNA that's going to match up is going to be C, A, U, but we don't look at the anticodon. What we look at is the mRNA strand. Oopsies, I missed one here, excuse me. It's going to be AUG, will be my tRNA. Sorry about that. So A is in the center right here. I'm going to look at this A, and the one of the 4 bases that comes next will be this U, and one of the 4 bases that comes next will be this G. And so that corresponds with the amino acid methionine. Alright, so we know that amino acids correspond with the mRNA codon. So this is how the process works of making proteins from the mRNA strand. Our ribosome can only fit 2 of these complexes in it at the same time. So while these 2 complexes are in it, our amino acids are nice and lined up next to each other, right? So what happens is these amino acids will connect. They'll kind of hold hands and branch out like this. Well, once they've connected, there's really no need for this tRNA complex down here, right? And so what will happen is that amino acid will break off from that tRNA. Now this tRNA isn't really needed, and so the RNA will poop out that tRNA basically. So now we have 2 amino acids put together. Now our ribosome can move over 1 more spot, and now we're going to have another tRNA. And so now this amino acid and this amino acid are going to connect. This one isn't going to be needed anymore, and so that one will be pooped out. And eventually what's going to happen is we're going to have a chain of amino acids that's going to be pooped out. That chain of amino acids is the protein. This is the important thing. That protein is what makes up almost our entire bodies, right? As living organisms, we're made primarily of proteins. So our eyeballs are made of proteins, our liver's made of proteins, our lungs are made of proteins, our hair is made of proteins, right? Almost everything we're made up of, including our characteristics, our hair color, our eye color, our feet size, are because of proteins. So our characteristics, the things that make us who we are all because of DNA. We start with a strand of DNA. That DNA is made into mRNA. That mRNA moves from the nucleus to the ribosome. And that strand of mRNA combines with tRNA carrying amino acids. Those amino acids combine with each other because of that code, and these amino acids make up proteins that are specialized to be certain characteristics and to do certain things inside of your body. So overall, that's transcription and translation, and I hope that helped. If, as always, you have any questions, let me know. Thanks, guys.